Hello viewers, we are going to look into the introduction of cell culture. What is a cell culture? An animal cell culture is the growth of cells in a favorable, controlled artificial environment such that we can make use of the cells that we are culturing. This concept of isolation of cells from the original tissue and therein maintaining them started very early in the 20th century. The concept and the techniques used in animal cell culture is a major tool in life sciences. It has provided a system for basic cell research, toxicity testing, gene therapy, and development of disease model, uh, monoclonal antibody production, and it has been a major contributor in vaccine development. Something of a brief history. Very early, it was initially started for understanding the concept of a nerve cell growth and development. First of a kind started in 1907, and a large-scale industrial scale establishment started in 1950, majorly due to the epidemic of polio. When we talk about how we are going to get these cells, what is the source of these cells, there are three major options, so to say. One is organ culture, another one is primary explant, and then cell culture. When we say organ culture, we are actually taking the organ or partially developed adult organ, most probably it will be embryonic organ, and we are culturing them and maintaining them for a considerable period of time. But the problem here is the organ needs proper nutrition, everything, even though we'll not be able to culture them or maintain them for a very extended period of time. When that particular experiment is over, we have to move to sourcing another explant. So from explant, from organ to organ, there's going to be variability. So because of which there was a second set of sourcing that was developed, which is called as a primary explant, wherein from this organ that has been maintained in a culture environment, a small sample will be taken, and this explant will be used for further maintenance of the cells as a three-dimensional part. But once again, here what happened was the growth of the cell was very slow, as well as the explant was not able, we were not able to manipulate them to the extent that we wanted. So thereby cell culture was developed. Cell culture is basically the cells that we collect from organs or primary explant, and we maintain them as a single cell status, either in an adherent form or suspended form. These cell cultures can be of three types. The cells that we are culturing in cell cultures can be of three types: precursor cell culture, differentiated cells, or stem cells. When we say precursor cell culture, we are talking about cells that have been sourced, and they can be modified or manipulated to become a particular type of cell. They are in some kind of a precursor state. They are not completely differentiated, neither are they in a stem cell state. Then we have the differentiated cells, wherein they are completely one particular type of cell. They are destined to be only that type of cell. Then the third option is the stem cells, wherein pluripotent stem cells can be maintained, and from them different types of cells can be uh, uh, manipulated and obtained through the hormones and the supplements we provide in the growth medium. Cell culture system, as we speak, it is an in vitro model system. There's nothing happening inside a living body. That is why it is called as in vitro, and the name cell culture or tissue culture is being used. So these cells are either anchorage dependent or anchorage independent cells, meaning they need a substratum to attach and grow. When that is the case, they're called as anchorage dependent cells. Most of the cells are of anchorage dependent type, because for them to settle, establish an anchorage and to become a monolayer, they need some kind of a substratum. So second type is anchorage independent, wherein they are generally their characteristics is not that they need a substratum to attach and grow. Those are called as suspension cell culture. When we take the cells from an explant and we establish a single genetically identical set of cells, that is clones, we are establishing a cell line from where they will grow into the culture dish, whichever dish we are using, form a monolayer if it is an anchorage dependent cells. If it is a suspension, they will reach a particular density, after which there needs to be changing of the media, and there needs to be dilution of the density of the cells. When we do that, when we subculture the already established cells, or otherwise called as passaging, they become the secondary cell culture. Okay. So in the types of cell lines, what we have is called as finite cell lines and infinite cell lines. Finite cell lines, as the name suggests, they have particular number of passaging they will survive, after which their survival becomes questionable and their morphological and functionality also changes. When we talk about infinite cell lines, we are talking about transformed cell lines. Either we are talking about cancer cell lines or non-cancer cell lines which have been transformed such that they have an inbuilt capacity to be immortal, meaning they can be established as a continuous cell culture wherein the number of passaging is not limited to. For example, most of the common cell lines that we have used, it's been used for a long time, are transformed human embryonic kidney, HEK293, epithelial cells from monkey, which is called as Vero cell lines, mammary cancer cell line called as MCF7, mouse fibroblast cell lines 3T3. See, this is an example of different cell lines. The first image shows the breast tissue. MCF10A is a normal cell, and after which you have MCF7, T47T, MD, MB231. All the three are different types of cancer cells. You can see the change in the morphological characteristics of the cell lines. In the second row, we have cervical cells, the first one being the normal cervical cell, and the next three options are cancerous cervical cells. And the third, if you see, these are all lung cells. You can see how drastically the morphology of the cells have changed, and thereby the functionality of which will also be majorly impacted. When we talk about the purpose of developing or using cell lines even after so much of technological advancement, is that for the production of antiviral vaccines we need a lot of recombinant therapeutic protein development, a lot of testing has to be done. um, uh, Even for biopesticide based uh, development for testing and for control, baculovirus based biopesticide has been brought up, and since it is a viral based pesticide, you need a cell for the virus to attach themselves and grow, to multiply in number. So even for that purposes, the cell lines are used. As we have already seen, monoclonal antibody production, and there's something called as microfluidics organ on a chip has been coming up lately, wherein the cells are being manipulated in such a way that each in a three-dimensional setup they are being made into. For example, if you're talking about cells, when we are going to add cells in a medium, we have discussed something called as anchorage dependent, where we are saying that the cells are going to go attach itself to a substrate and to grow. They are growing, but to form a monolayer, the cells just growing is not enough. There needs to be some kind of an extracellular matrix, that is protein complex, that needs to be produced such that these cells form a layer. They are attached or stuck to one another in such a way that there is some form of a communication between the cells, toxin removal, and two-dimensional structure that is being built. In the cell culture technique, as of now, only two-dimensional is seen. So scientists are developing a three-dimensional microfluidic system wherein there is movement of components from one cell to another, along with the communication, the fluid as such, how it is going, all the components that are moving, how each cells respond to the stimulus from the other cell. All these kind of things are being developed. It's been there for quite a long time, and a lot of advancements have been recorded so far. Even though this has been the cell culture technique or cell culture has been there for a long time, still there are a lot of ethical considerations around it, and major being the biohazard. Whatever waste that we are producing through cell culture techniques has to be handled as a biohazard and properly disposed of. There are a lot of legislations and guidelines around the procurement, use, and disposal of all the components related to cell culture, right from the media components, all the substituents that are added in the media, the cells that are purchased, and particularly there's something called as a serum that is being added in the media. The serum usually is fetal bovine serum or fetal calf serum, which is a major ethical consideration among animal welfare units because it is obtained from a fetal tissue. So this is the basic introduction, and in further videos we look into how and when this all started, with what particular cell line cell culture technique was started. Thank you.